Dear participants, hello, good morning, all of you once again. Hereby we are going to start the second class, that is biotechnology and its application. So the participants who have joined right now, so just quickly tell me, am I audible? Is the screen visible to all of you? Just text me in the chat box. The participants, can you hear me? Am I audible? Am I audible? Let us wait for another 2 minutes for other participants to join. Okay. Okay, so hello all again, once again to all the participants. I am Debasis Sahoo, lecturer in Botany, and today I am going to take this class, that is biotechnology and its applications in the field of medicine. You are watching DHSC online academic support program for the NEET and JEE aspirant students. This is sponsored by Government of Odisha, DTC Government of Odisha. So let's quickly begin the second class, which is on topic the applications of biotechnology in the field of medicines. Now, as you can see in the screen, here we are going to talk about the applications of biotechnology in the field of medicine, in the field of pharmaceutical industry, and how this biotechnology help us to get back from all those various diseases and all. ପିଲାମାନେ ତମେ ଏବେ recently କୋଭିଡ ବିଷୟରେ ଶୁଣିଥିବ 2 years 3 years back କୋଭିଡ ଯିଏକି ଆସିକି ଗୋଟେ pandemic ହିସାବରେ ଆସିଥିଲା and ବହୁତ ସାରା କ୍ଷୟକ୍ଷତି କରିକି ଯାଇଥିଲା and ସେଇ ଯୋଉ କୋଭିଡ ଭାଇରସ ସହିତ ଲଢିବା ପାଇଁ କି ଆମେ vaccine ର development କରିଥିଲୁ ନା ଏଇ ଯୋଉ formation of vaccine ହେଇଥିଲା that vaccine is not made by doctor ମାନେ ତ utilize କଲେ the vaccine is made by the scientists and those scientists who are basically in the field of biotechnology ସେମାନେ ପାର୍ଟି ଡେଭଲପ କରିଥିଲେ and ସେଇ biotechnology ମାନଙ୍କ development ରେ ଏ ଯୋଉ vaccine formation ହେଇଥିଲା ତାକୁ ଆମେ utilize କରିକି ଆମେ ଯୋଉ novel corona virus main COVID-19 ଯୋଉ ଥିଲା ତାକୁ ଆମେ tackle କରିପାରିଲେ and ଆମେ ନିଜକୁ ତା କବଳରୁ ବଞ୍ଚେଇ ପାରିଲେ ଠିକ ସେମିତି ଯାହା ବି କିଛି diseases ତମେ ଦେଖୁଛ ଏବେ recently ଆମର ICMR Indian Council of Medical Research ଆମର ଏଇ ଓଡିଶାରେ ଯୋଉ RMRC ଅଛି ରିଜନାଲ ମେଡିକାଲ ରିସର୍ଚ୍ଚ ସେଣ୍ଟର ଲାବୋରେଟୋରୀ ଯୋଉ ଅଛି ସେଠି ମ୍ୟାଲେରିଆ ଭ୍ୟାକ୍ସିନ ଫର୍ମେସନ ହେଇଛି and ଯୋଉଟା କି ବହୁତ ଜଲ୍ଦି ହ୍ୟୁମାନିଟି ଡେଡିକେଟ କରାହବ ବହୁତ କମ ପଇସାରେ ଇଟ ଇଜ ଇଣ୍ଡିଜିନିୟସ ଭ୍ୟାକ୍ସିନ ଆମ ଓନ୍ କଣ୍ଟ୍ରି ଆମ ଓନ୍ ନଲେଜ ଆମେ ଏହାକୁ ବନେଇଛେ ତ ଏଇ ଯୋଉ ଭ୍ୟାକ୍ସିନ ଫର୍ମେସନ ହେଇଗଲା ବା ଏଇ ଯୋଉ କୌଣସି ଡିଜିଜ କୁ ଡାୟଗ୍ନୋସିସ କରିବା ହେଇଗଲା ଏସବୁ କେସରେ ୱି ନିଡ ଦ ହେଲ୍ପ ଅଫ ଦିସ୍ ବାୟୋଟେକ୍ନୋଲୋଜି ତ ଆଜି ତମକୁ କ୍ଲାସ 12 ର ଏଇ ଚାପ୍ଟରଟି ଅଛି ବାୟୋଟେକ ର ଆପ୍ଲିକେସନ the application part, let us quickly know about how this biotechnology is helping us in preventing these various kind of diseases and how it is helping us to diagnose a disease. Eleven English there is a saying that prevention is better than cure. So the cure କରିବା ଅପେକ୍ଷା if you can prevent it, ସେ ରୋଗ ହେଲା ପରେ ତାକୁ କ୍ୟୁର କରିବା ଅପେକ୍ଷା if you can prevent it, then ଏଟା ଗୋଟେ ବହୁତ ଭଲ ଷ୍ଟେପ୍ ହେଇପାରିବ। बट सपोज ଆମେ ପ୍ରିଭେନ୍ସନ୍ କରିପାରିନେ and ଆମକୁ ସେ ଡିଜିଜ୍ ହେଇଯାଇଛି, ବଟ୍ ଏବେ ବି ଆମ ପାଖରେ ଟାଇମ୍ ଅଛି ଡିଜିଜ୍ କୁ ଠିକ୍ କରିବା ପାଇଁ କି ଆଉ ସେ କ୍ୟୁର କରିବା ପାଇଁ କି ସବୁଠୁ ବଡ ଷ୍ଟେପ୍ ଆଉ ସବୁଠୁ ଫର୍ଷ୍ଟ ଇନିସିଆଲ ଷ୍ଟେପ୍ ହଉଛି ଡାୟଗ୍ନୋସିସ୍। If we can diagnose a disease earlier, then it is going to help us in a long term. For example, बहुत टाइम हम मार्क करथिबो whenever we fall ill, ଆମେ ଜରରେ ପଡିଥାଉ तो we go to see a doctor and the doctor हमକୁ देखंदी से doctor recommend करथांदी किछि चेक ପାଇଁ कि like the blood test, the urine test, the sputum test, इस टेस्ट ପାଇଁ हमको अलग अलग ये टेस्ट सब करके आसो then अच्छा जब हम टेस्ट करूछे और टेस्ट और रिपोर्ट अनुसार से हम डायग्नोसिस करछंती बट माय पॉइंट इज कि टेस्ट जो रिजल्ट जहां भी आसुची the already the disease ହେଇଛି ବୋଲି ଏଟା proof the disease ହେଇ ସାରିଲାଣି but ଆମେ ଯଦି prevent କରିବାକୁ ଚାହିଁବା ତାହେଲେ ଆମେ earlier part ଟା ଦେଖିବାକୁ ପଡିବ ତ ଯଦି ଗୋଟେ bacterial infection ଟି ହେଇଛି ମୋ body ରେ ଆଉ ସେ blood ରେ ଯାଇକି ମିଶିଛି that means already I was infected with the bacteria ସେ bacteria ମୋତେ infection କରି ସାରିଲେଣି but ଏମିତି କୋଉ ଟେଷ୍ଟ ଅଛି ଯାହା ଦ୍ୱାରା ମୁଁ ଆଗରୁ ପ୍ରିଭେଣ୍ଟ କରିପାରିବି ଆଗରୁ ଜାଣିପାରିବି तो ता ପାଇଁ ଆସିଲା ଆମର ବାୟୋଟେକ୍ ର PCR polymerase chain reaction ବା ELISA ELISA enzyme linked immunosorbent assay so these are the basic molecular diagnostic tool ଯାହାକୁ ତିଆରି କରା ହେଇଛି ବାୟୋଟେକ୍ନୋଲଜି ବାଲାଙ୍କ ଦ୍ୱାରା for the service to humanity ସେଇ ପାର୍ଟ ଉପରେ ଆଜି ଆମେ ଡିସ୍କସନ କରିବା ଏଇ ସେକେଣ୍ଡ କ୍ଲାସରେ in the morning class in the very first class we have discussed about the role of biotechnology in the field of agriculture. And in the second class, we are going to discuss the role of biotechnology in the field of medicine. So I'm requesting all the participants to kindly stay till last of the class. There is a small question answer segment at the end of this class. So many class को attend करूछो पढ़ुछो सुनो end moment नहीं से मने रुह and then a question answer round participate करो and enhance your knowledge which is going to help you in your upcoming NEET entrance examination. Clear? Okay. So without wasting any time, let's quickly move on to the next slide. That is the applications of biotechnology in the field of medicine. See, dear students, the recombinant DNA technology, or shortly called as RDT, it helps for the mass production of safe and more effective therapeutic drugs. You know, biotech को हम फर्स्ट मेजर स्लाइड देखथिले कि ये हेल्प करते थेराप्यूटिक्स ड्रग्स फॉर्मेशन करिया ପାଇଁ कि ड्रग्स मींस इट मेडिसिन कथा पड़ जाउची जो फार्मा इंडस्ट्री जो मेडिसिन सब तैयारी करा होची से ड्रग्स थेराप्यूटिक ड्रग्स जो फॉर्मेशन करिया रे आमे बायोटेक रे हेल्प होचे द प्रोडक्ट्स फ्रॉम नॉन ह्यूमन सोर्सेस induce unwanted immunological responses but recombinant therapeutic does not have such problems যদি আমি কিছু নন হ্যুমান সোর্স কি জিনিসকে আমি ইনডিউস করুছে আনুছে যদি ଆମ ବଡି ଯାଇ କଣ କରୁଛି ନା ଅନୱାଣ୍ଟେଡ ଇମ୍ୟୁନୋଲୋଜିକାଲ ପାର୍ଟ କୁ ସେ ଅବଲେ ବଢେଇ ଦଉଛି ଏକ୍ସାମ୍ପଲ ଦେଇ କହୁଛି ଯେମିତି କି ହେଲା ଇନସୁଲିନ ୟୁ ଅଲ नो ଇନସୁଲିନ ଇଜ ଅ ବେସିକ ହରମୋନ ଆମ ବଡିରେ ପ୍ରଡ୍ୟୁସ ହେଇଥାଏ ସେଇଟା what is the role of insulin to regulate the sugar ଆମ ବଡିରେ ଯୋଉ ସୁଗାର ପ୍ରଡକ୍ସନ ହଉଛି ଆମେ ସୁଗାର ପ୍ରଡକ୍ସନ ତ କଣ ଆମେ ଯୋଉ we eat food from that food the food is digested the digestion will simplified sugar that is called as glucose now it is the role of the blood to carry this sugar to carry this glucose to all the cells now remember the mechanism this glucose is basically a polar molecule ठीक है it is hydrophilic in nature so water soluble अछि but our जो cell ର membrane अछि it is semi permeable in nature and what do you mean by semi permeable that means it has selectively permeability yes or no that means it will allow only those molecules selectively allow those molecules which are required or which are not required से basis से को allow कर तो जो semi permeable membrane अछि it is made up of phospholipid bilayer this phospholipid bilayer will not allow each and every molecule to come inside and also to go outside. So the glucose is present in the blood but the glucose needs to transfer into the cell. The glucose जे blood रे अच्छी से अभी cell को जीवा को चाहूँगी એટલે ସେ cell को entry କରି पारुनी because मझिरे आसीगला कि मझिरे is present ହେଇଛି cell membrane or plasma membrane which is selectively permeable in nature. Now in this case what will happen, this insulin hormone will come and it will open the transporter, glucose transporter GLUT, a channel protein member for the transportation of glucose from the blood to the cell. Clear? तबे बुझो जिनस ओडिया रे कहूची बुझो पार्ट टा कोन कहयो जउची कि आमे खाइबा खाइलु खाइबा जाय कि हम बॉडी रे डाइजेस्ट होला पर से ग्लूकोज रे कन्वर्ट हे गेला सिंप्लिफाइड शुगर रे स्मॉल इंटेस्टाइन से अभी अब्जॉर्ब हो गेला हम ब्लड वेसल्स से पड़ेला. नाउ ब्लड बीइंग द मोबाइल कनेक्टिव टिश्यू, the blood entire body रे से फ्लो करछि सर्कुलेट करछि तो ब्लड एबे सब सेल बना पाखर जाकी पहुंची जे कैरिंग दिस ग्लूकोज. बट से जो ब्लड रे ग्लूकोज टा सेल भितर से तीन बेले जाई परिबो जेबे ता पाखरे इंसुलिन नामक हार्मोन दी अछि. तो इंसुलिन इज अ हार्मोन जहाँ को बनैछि हम बॉडी रो गोटे ग्लैंड अछि एंडोक्राइन ग्लैंड सबुठु लार्जेस्ट एंडोक्राइन ग्लैंड दैट इज कॉल्ड एज पैनक्रियाज. और से पैनक्रियाज रो आइल्स ऑफ लैंगरहैंस रो बीटा सेल्स से माने इंसुलिन प्रोडक्शन करथन. तो ए इंसुलिन जो अछि से बे आसिला तार प्रेजेंस रे अब कोन ये जो सेल पाखे गोटे मेम्ब्रेन पाखे गोटे ग्लूट ट्रांसपोर्टर अच्छी ग्लूकोज ट्रांसपोर्टर चैनल टी अच्छी तो चैनल टी ओपन हो गया. इट्स लाइक इंसुलिन इज द की व्हिच ओपन द ग्लूट ट्रांसपोर्टर ग्लूकोज ट्रांसपोर्टर से ओपन चैनल को ओपन कर दो एंड नाउ द ग्लूकोज विल मूव इनसाइड द सेल एंड इन दिस वे ब्लड रे ग्लूकोज लेवल टा रेगुलेट हे पाउची. जो बड़का ग्लूकोज रही गला लेट्स मीन सेल को जेतिक अमाउंट ग्लूकोज दरकार जेतिक अमाउंट ग्लूकोज मिली गला ता बाद रे जो ग्लूकोज बळी पड़िची से ग्लूकोज को हम इंसुलिन कन्वर्ट कर दोसला कोथरे ग्लाइकोजन. then the plant produces glucose and store in the form of starch. ठीक सेम हम बॉडी रे जो ग्लूकोज हमें खाई की पाई छे से ग्लूकोज को इंसुलिन कन्वर्ट कर दोसला ग्लाइकोजन रे एंड इन दैट वे द ग्लूकोज इज वेरी बेसिकली बैलेंस्ड इन द बॉडी. हम बॉडी रे से ग्लूकोज टी बैलेंस हो गला. क्लियर? ये वाला मैकेनिज्म. तो एक काम पई की रिस्पांसिबल के इंसुलिन बट जदि आम बॉडी रे इंसुलिन सफिशिएंट अमाउंट ऑफ प्रोडक्शन हे पारुनी तीनटा पॉइंट मन रख. इफ द इंसुलिन इज नॉट प्रोड्यूस्ड इन द बॉडी बा इंसुलिन तो बनसा पैनक्रियास कोन कारण बसबेट म्यूटेशन हे गेला किछि चेंज हे गेला पावर कट हे किछि भी हेछि इंसुलिन इज नॉट प्रोड्यूस्ड इंसुलिन बनुनी. नंबर 2, इफ द प्रोड्यूस्ड इंसुलिन इज नॉट सफिशिएंट जितने की बनेला तो सफिशिएंट है नहीं या तो बिल्कुल बनुनी कौन सी डिफेक्ट है कि नहले जितने की बनू दैट इज नॉट सफिशिएंट दैट इज नॉट एनफ. थर्ड रीजन होची जो इंसुलिन बनुची से डिफेक्टिव अछि. देखो ना या तो बिल्कुल बनुनी या तो कम बनुची या तो डिफेक्टिव बनुची और लास्ट हैला जोटा जोटा सबुट इंपोर्टेंट पार्ट ता जदी बनुथिबा इंसुलिन को हम बॉडी रेजिस्ट करि जाउची हम बॉडी रे रेजिस्टिविटी अछि ना बारंबार सेम जिंस से यूटिलाइज करले रेजिस्टेंस पड़े आसो तुमको सब दिन सकाळ उठनी गोटे प्रकार आइटम देबा खाया पाएंगे. लेट्स से यू लव बिरयानी द मोस्ट and every day in the morning in the lunch in the dinner breakfast lunch dinner खाली बिरयानी दिया हबो धीरे धीरे तुम बिरयानी प्रति गोटे घुणा हबा आसियो लाइक तुम से जो रेजिस्टेंस आसियो बिरयानी को नहीं कि you want normal food you want some other food as compared to बिरयानी. तो सब दिन सेही टाइम रो डाइट तुम फॉलो करिवो जदी तुम आगो जाएगी ता प्रति गोटे खराब भावना पड़े आसियो देन कहीं हम डाइवर्सिटी दरकार अछि. तो इट इज इन द इंसुलिन जो बॉडी प्रोड्यूस करछि एंड लॉन्ग टर्म से इंसुलिन टे कौन रेजिस्टेंट दे देछि हमर किसी सेल्स इंसुलिन को मान नाही. तो ए जो चारटा केस सिनेरियो तुम आगे देली कि इंसुलिन प्रोडक्शन होनी बा इंसुलिन कम प्रोड्यूस होछि बा इंसुलिन का डिफेक्टिव प्रोड्यूस होछि बा बॉडी ताको रेजिस्ट कर देछि. ए केस रे वी नीड इंसुलिन फ्रॉम द अदर सोर्सेस. अब बाहर इंसुलिन नबाको पड़िबो बिकॉज़ विदाउट इंसुलिन हम बॉडी रे ग्लूकोज मेटाबॉलिज्म हे परिबो नाही ग्लूकोज को रेगुलेट हम करि परिबा नाही. और यदि ग्लूकोज बढ़ी की रहिला सुगर बढ़ी की रहिला हाइपरग्लाइसेमिया ताको हम कहबा व्हिच इज रिफर्ड टू एज डायबिटीज मेलाइटस. यू ऑल नो इंडिया रो घरे घरे डायबिटीज अछि. इंडिया इज द डायबिटीज कैपिटल ऑफ द होल वर्ल्ड. तो देखो 196 कंट्री विच इंडिया इज द ओनली कंट्री व्हिच इज इन द टॉप in case of these diabetes, in case of this obesity, each of these India have a top equivalent. Then which is not a credit for our country. ये हम अपनी कोई खुशी का कथा नहीं. ଏବେ ଯୋଉ ଡାଇବେଟିସ୍ ଏତେ ହଉଛି ଡ୍ୟୁ ଟୁ ଆवर ସେଡେଣ୍ଟାରୀ ଲାଇଫ ସାଇକେଲ। ଆମେ ଯୋଉ ଖାଉଛେ ପିଉଛେ ଗୋଟେ ଜାଗାରେ ବସୁଛେ, ସେଡେଣ୍ଟାରୀ ଲାଇଫ ସାଇକେଲ ଯୋଉ ହଉଛି, ଆମ ଏନଭାୟରନମେଣ୍ଟ ଯେମିତି ଚାଲିଛି, ତାପରେ ବି ଯୋଉ ଜିନ୍ ଯୋଉ ମ୍ୟୁଟେଟ୍ ହଉଛି, ଏଇ ସବୁ କେସରେ ବହୁତ ସାରା ହଇରାଣ ଆମେ ହଉଛି। ନା କମିଂ ଟୁ ଟପିକ, ଏ ଟପିକ ଆମେ କଣ ଦେଖୁଛେ ଏଠି କି ଯୋଉ ନନ୍ ହ୍ୟୁମାନ ସୋର୍ସ ରୁ ଯେତେବେଳେ ଆମର ଏବେ ଇନସୁଲିନ ପ୍ରଡକ୍ସନ ହେଇପାରିଲାନି, ତ ଇନିସିଆଲି वी आर डिपेंडेंट अपॉन द स्लॉटर हाउस। यू नो स्लॉटर हाउस कसेई खाना, ଯୋଉଠି ଅବିକା ଗାଈ କଟା ହୁଏ, ଯୋଉଠି ଆଗେ ଛେଳି କଟା ହୁଏ, ଘୁଷୁରୀ କଟା ହୁଏ, ସ୍ଲଟର ହାଉସ। ଯୋଉ ପିଗ ରୁ, ସେ କାଉ ରୁ, ସେ ବୁଫାଲୋ ରୁ ତାଙ୍କ ବ୍ଲଡ ଇନସୁଲିନ କୁ ଆଣିକି ଆମ ବଡିରେ ଇନସର୍ଟ କରାଯାଇଥିଲା, ଯୋଉଟା କି ଟେକ୍ନିକାଲି ତ ଇନିସିଆଲି କିଛି କାମ ପାଇଁ ଠିକ ଥିଲା, ବଟ ଆଗକୁ ଯାଇକି ଲଙ୍ଗ ଟର୍ମ ଇଫେକ୍ଟ ପକେଇଲା, ବହୁତ ସାରା ଇନଫେକ୍ସନ ହେଲା ବଡିରେ, ଆଉ ଯୋଉଥିପାଇଁ କି ଆମେ ସେଇ ଯୋଉ ନନ୍ ହ୍ୟୁମାନ ସୋର୍ସେସ ଉପରେ ଡିପେଣ୍ଡେଣ୍ଟ ରହିବାଟା ବି ଅସୁବିଧା ହେଲା। ତ এই লাইনটা ভলসে পড়, বুঝো কোন পয়েন্ট কইলা এটি না, the products from the non-human sources induce the unwanted immunological responses। जो इम्यूनोलॉजी रेस्पॉन्स हम दरकार नहीं से पार्ट रखो, अनवांटेड पार्ट रखो, बॉडी में भी एनहांस स्टार्ट हो जाए। But recombinant therapeutics does not have such problems। ତ ଆମେ ସବୁବେଳେ କମ୍ପେୟାର କରୁ ନା, ଆମେ କୋଉ ଜିନିଷକୁ ଗୋଟେ ଆଣୁଛେ, ଆମେ କମ୍ପାରିଜନ କରୁଛେ କି ଆର ପ୍ରୋଜ କଣ, ତାର ପ୍ରୋଜ କଣ, ଆର କନ୍ସ କଣ, ତାର କନ୍ସ କଣ, what is the merit, what is the demerit। ଆମେ ଯଦି merit demerit କୁ ଚେକ୍ କରୁଛେ, ଆମେ ଦେଖିପାରିଲେ କି the product from the non human sources, ଯୋଉ ପ୍ରଡକ୍ଟ ଆମେ ନନ୍ ହ୍ୟୁମାନ ସୋର୍ସ ରୁ ଆଣିଛେ, ସିଏ କଣ କରୁଛି ନା ଏନହାନ୍ସ କରୁଛି ଅନୱାଣ୍ଟେଡ ଇମ୍ୟୁନୋଲଜିକାଲ ରେସ୍ପନ୍ସ, while the recombinant therapeutics does not have the problem। तो आप अगर से प्रॉब्लम किसी देखिएको बोले which is a good thing, ये प्रॉब्लम किसी आप अगर नहीं। then at present about 30 recombinant therapeutic drugs have been approved, and of these 12 are being marketed in India। तो ये जो डाटा मन रखिबा हम पाखे वर्तमान 30 टा थेराप्यूटिक ड्रग प्रेजेंट अछि, रिकॉम्बिनेंट therapeutic drugs are present, and among them 12 are marketed and approved in sale in India। क्लियर, ये थेराप्यूटिक जहां भीतर इंसुलिन गोटे प्रमुख ହେଇକି ରହିବ। तो एवढा जिंस just remember in this slide that recombinant technology helps in the production of mass production of safe and more effective, ଏଇଟା word ବହୁତ ଭଲ ଜିନିଷ, safe and more effective therapeutic drugs production से help କରୁଛନ୍ତି किए ना, this RDT recombinant DNA technology helps for the mass production of the safe and more effective therapeutic drugs। The products from non-human sources induce unwanted immunological responses, but recombinant therapeutics does not have such problems। You need to remember what are the pros and what are the cons। ପିଲାମାନେ ବୁଝିପାରିଲ? Any doubt till now? Dear students, if you have any doubt, then kindly tell me in the chat box. We'll go and we'll proceed on this. Now, see, so what are the applications of biotech in the field of medicine? You can see it is used for the genetically engineered insulin, it is used in gene therapy, and it is used in molecular diagnosis। GGM on the genetically engineered insulin, that is the humulin बोली कहूचे। What is human insulin, which is produced inside the body, inside the pancreas, inside the isles of Langerhans, inside the beta cells, and something which is produced outside the human body in a bacterial cell, in a E. coli cell, that is called as genetically engineered insulin। Then the a production ने हमें केमती help नो जो application है बायोटेक्नोलॉजी देखिबा। Second, we will focus on gene therapy। So what is gene therapy? Gene therapy कौन होची ना, हमें गोटे faulty gene, गोटे defective gene को replace करूचे by a healthy and a functional gene। You know, in principles of inheritance and variation, I have taught you one thing, that genotype controls phenotype। Remember this term? Genotype controls phenotype। So what does exactly it means? Then genotype जो है our internal appearance, our genetic structure, it basically controls or regulates our external appearance, our phenotype। मो हाइट केते हबो, मो वेट केमती हबो, मो स्किन ना कलर केमती हबो, मो हेयर टेक्सचर केमती हबो, all these things are being governed by the gene which I have inside me, and or which I have inherited from my parents। Yes or no? तो मो पेरेंट्स को जो जीन मो पाखु आसी से ड्यूरिंग द कोर्स ऑफ इनहेरिटेंस जो जीन मो पाखु आसी जो जीन मो पाखे एक्सप्रेस होची से डोमिनेंट जीन से एक्सप्रेस जीन मो आगु जाई कि मोर फिनोटाइप को कंट्रोल करची what will be my shape of the body, what will be my height, what will be my color, what will be my blood group. All these minute things are governed by my genes only. Yes or no? So genotype control phenotype. Suppose there is a basic problem in the gene and the gene has a problem सके से आगे जाएंगे से organism में बहुत सारे defect देखने को मिलती. So to cure it or to permanently cure this, हमको ये कौन करेगा पड़ेगा gene manipulation करेगा पड़ेगा. We need to alter in the gene. We need to do some modifications, some alterations in the genetic level. तापर से जो जीन अच्छी जो तापर जो लैंग्वेज अच्छी ए यू जी सी जो कोडन सब अच्छी से टी जी सी ए यू जी सी कोडन को हमको मैनिपुलेट करियो पड़िबो मॉडरेट करियो पड़िबो एंड हमको से जो डिफेक्टिव जीन को आइडेंटिफाई करिके ताको रिप्लेस करिबा गोटे नॉर्मल गोटे फंक्शनल गोटे हेल्दी जीन तो कहवाटा बहुत सहज पिला माने बट आपको प्रैक्टिकली करिबा laboratory case रे करिबा is highly costlier expensive और तासुरा टाइम टेकिंग क्लियर तो तमे खाली आर पार्ट रे तो बेसिक क्वेश्चन जाने व्हाट इज जीन थेरापी कोठा का स्टार्ट हेतिला केमरे काम हेतिला ए पार्ट मन हेतिला फॉर अपकमिंग नीट एंट्रेंस एग्जामिनेशन हो या एनुअल हायर सेकेंडरी एजुकेशन एग्जाम जो एग्जामिनेशन हो तो video case today you are going to expect this kind of 2 mark question or 1 mark question from this chapter so समस्त को रिक्वेस्ट करूची कि क्लास को एंड जाए कि लर्न करा देखो आ से 2 मार्क क्वेश्चन को मन रहो कोन लिखवार अछि बोली and last असला molecular diagnosis से we are basically reliable upon the PCR the ELISA technique PCR technique से ऊपर हमें देखो in today's class I am going to deal with the first two but first the genetical engineer insulin and second is the gene therapy then the part details se any doubt at any particular time you can always ask me in the comment box or in the chat box I am there to answer each and every queries then okay, so without wasting any other time, let's quickly jump into the genetically engineered insulin part. Okay, so you can see here, insulin is used to manage adult onset diabetes. Then there is a pathway I'll explain you in details. Then insulin from the pancreas of animals like the cattle, the pigs, this causes basically what? Allergy or other types of reaction to the foreign protein. ठीक है ये जो फॉरेन प्रोटीन हमें रिसीव करिछे कोठो फ्रॉम द cattle or the pig basically from the slaughterhouse it causes allergy you know any kind of foreign particle entering to our body is called as antigen and if that antigen is able to do any kind of disease it is called as pathogen we are the host that is the pathogen जैसे हम बॉडी किसी allergic reaction करछि allergen ता प्रकार जेता हमें allergy बोली कहउछे and other type of reaction to the foreign protein ये सब problem थिला next now it is possible to produce human insulin using bacteria this is the role of this is the application of biotech in the field of medicine that is it is possible to produce human insulin जोटा हम बहुत दरकार हमको नीड बहुत अच्छी से जिनसटी कारण हमर जो सेडेंटरी लाइफ साइकिल जो रेट चली छी जो डायबिटीज मेलाइटस हमको बहुत अफेक्ट करछि इंडिया अबिका ग्लोबल कैपिटल हिस्सा डायबिटीज मेलाइटस रो तो हमको इंसुलिन बहुत आवश्यकता अछि और इंसुलिन अर्लियर हम पाउथिले गो टू स्लॉटर हाउस लाइक फ्रॉम कैटल्स फ्रॉम पिग्स बट सीए हमको अनवांटेड थिंग्स भी दउथिला जेमति कि एलर्जी भी ता सहित आस्तला हमको हैरान होबा पड़थिला बट नाउ जदी हमें से जो इंसुलिन को गोटे बैक्टीरिया टू बनै पड़छे दैट इज एन अचीवमेंट आवर जिनसटी हमको आगो जाई हेल्प करले so you can see is here is the human cell जेहेतु human is an animal तो animal cell basically circular थाए this cell जेहेतु human गोटे eukaryotic तो eukaryotic organism it has a definite nucleus definite nuclear membrane तो ये जो न्यूक्लियस भीतर अछि न्यूक्लियोप्लाज्म and inside that nucleoplasm chromosomes are there so it is a basically chromosome so ये जो chromosome रहिला it chromosome contains the gene you know cell inside the cell nucleus is there inside the nucleus nucleoplasms are there inside the nucleoplasm the chromosomes are there એટલે મને મન રાખ્યો chromosome we cannot see the chromosome all the time It is only visible during cell division and in particular the late metaphase and early anaphase. तो माइटोसिस रिएक्शन हुए हम बॉडी रे माइटोसिस जो है सेल डिवाइड करथाय सेल मल्टीप्लाई करथाय से टाइम रे हम देख परिबा कि मेटाफेज सरी आसुथिबो एनाफेज स्टार्ट हे आसुथिबो से पर्टिकुलर सेगमेंट रे वी आर एबल टू लुक एट द chromosomes. These chromosomes are basically condensed from inside the chromosome. Chromatids are there. Like these are the two sister chromatids. Yes or no? These are the two sister chromatids. This is the centromere, and these are called as two sister chromatids. Inside the chromatids, chromatins are there. If I go from nucleus रे आसिला न्यूक्लियोप्लाज्म ता भीतर अछि क्रोमोसोम भीतर क्रोमाटिड रे क्रोमाटिन आसिला हेटेरोक्रोमाटिन देन ता थोड़ा जदि आउट डिकंडेंस कर देखिबो यू विल गेट यूक्रोमाटिन आ जो क्रोमाटिन फाइबर अछि से क्रोमाटिन फाइबर भीतर को माने अछंती न्यूक्लियोसोम्स आर देयर. What is nucleosome? When 200 base pair of DNA is wrapped around a histone octamer, then the structure is called as nucleosome. Yes or no? ये basic molecular biology part तो मैं कहूँगी से part रे focus वाले सुनता. तो ये जो न्यूक्लियोसोम अच्छी से न्यूक्लियोसोम रे किए होला हिस्टोन ऑक्टामर रे चारिपटे किए रैप करछि डीएनए कॉइल ठीक अछि अगेन तो से डीएनए को नहीं देखिबे डबल हेलिकल स्ट्रक्चर डीएनए से डीएनए में जो स्ट्रैंड सब अच्छी जे स्ट्रैंड को हमें कहू जे जीन बोली कहू द फंक्शनल सेगमेंट ऑफ डीएनए इज कॉल्ड एज द जीन और से जीन भीतर जो इंफॉर्मेशन अछि दैट इज कॉल्ड एज जेनेटिक कोड्स जेनेटिक कोडन से जेनेटिक कोडन आगो जाई की कॉपी करके एमआरएनए फॉर्मेशन करछि और से एमआरएनए बाहर को जाई की राइबोसोम पाखरे पहुंचे की प्रोटीन फॉर्मेशन करछि. तो इंसुलिन इज अ प्रोटीन. To make this insulin, to make this protein, we need information, and that information is stored. The information is kept inside the DNA, inside the gene, and DNA is residing inside this chromosome. Yes or no? तो हमें कहले कौन ये हला गोटे ह्यूमन सेल ये हला गोटे बैक्टीरियल सेल बैक्टीरियम ये कहले बैक्टीरिया अछि. Now this bacteria is having a plasmid. बैक्टीरिया कौन अछि प्लाज्मिड अछि. Remember what is plasmid? Plasmid कौन हला एठी आसिला प्लाज्मिड गोटे बैक्टीरिया रो सेल रो ऑर्गेनेल्स ए जो ऑर्गेनेल टी जो सेल ऑर्गेनेल अछि प्लाज्मिड व्हाट इज अ प्लाज्मिड इट इज autonomously self-replicating. I'm requesting each and every participant to write it, to note it in your copies for your future reference. Autonomously self-replicating extra chromosomal double-stranded circular DNA. So what is the plasmid? Plasmid कौन है ला? It is autonomously self-replicating extra chromosomal double-stranded circular DNA. So basically, chromosome in bacteria there is nucleoid. There is no nucleus. There is nucleoid. So the nucleoid रे क्या है chromosome अच्छी. से chromosome को छाड़ देला परे तापर गोटे extra chromosomal DNA present है. That is called as plasmid. See autonomous body. It can self-replicate. निज निज replicate करि परथिबो. Extra chromosomal अच्छी. Double strand अच्छी. Circular DNA टी अच्छी. ए बड़ा दिखते हैं plasmid. Extra chromosomal circular double stranded DNA. This is a plasmid. तबे जिनस हला कोन we are having human cell we are having a bacterial cell आमे करिवा कोन human cell रु आमे स्लाइस कर देले से क्रोमोसोम को आमे से क्रोमोसोम को काढिले तार जो रिक्वायर्ड पार्ट ता अछि ताको आमे स्प्लाइसियोसोम देकी बाय रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम रा हेल्प रे आमे ता कट कर देले the gene which is responsible for controlling the formation of insulin आमे से पार्ट ता को कट कर देले कोथरे कट करछे by using restriction endonuclease enzyme RE. Restriction endonuclease enzyme जो है ला restriction enzyme जो है ला these are target specific the particular target को जाएंगे स्लाइस कर दीजिए स्लाइस करी कट कर दीजिए. नेक्स्ट हम कोन गले एपटो गोटे प्लाज्मिड ता को आइसोलेट कर लियो आइसोलेशन माने ताको बाहर को काढिबा देन डीएनए आइसोलेशन हुए तो एक्सट्रैक्शन प्यूरिफिकेशन देन आइसोलेशन आसिला जो रेस्ट्रिक्शन एंजाइम यूज करके हम आपको कट करते यूजिंग द सेम रेस्ट्रिक्शन एंजाइम हम आपको भी कट करिवा ऑलवेज रिमेंबर जो रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम हम यूज करते टू कट दिस क्रोमोसोम we need to use the same restriction enzyme to cut the plasmid. କାହିଁକି ଦିଟା କ ସମାନ ସମାନ ମାପର କଟା ହେଲେ ହିଁ ଏ ଯାଇ ତା ଜାଗାରେ ଆସି ଲାଇଗେଟ୍ କରିପାରିବ। ଫର ଏକ୍ସାମ୍ପଲ ମୁଁ ଗୋଟେ ଜାଗାରେ 2 ମିଟର ଗ୍ୟାପ ରେ କାଟିଛି ଆଉ ଗୋଟେ 2 ଇଞ୍ଚ ର ଗ୍ୟାପ ରେ କାଟିଛି। ଦିପଟେ 2 ସମାନ ଅଛି ହେଲେ ମିଟର ଆଉ ଇଞ୍ଚ ଅଲଗା ଅଲଗା ହେଇଗଲେ ଅସୁବିଧା ହେଇଯିବ। 2 ମିଟର ଯେତିକି 2 ଇଞ୍ଚ ସେତିକି ନୁହେଁ। କ୍ଲିୟର? ଦେନ୍ ଆମେ କଣ କଲେ ନା ଆକୁ ଆଣିକି ଏଇ ପାର୍ଟ ରେ ଲାଇଗେଟ୍ କରେଇଦବା। by using ligase enzyme ଆମେ ଜିନ୍ ଟାକୁ ଅବିକା ଇନସର୍ଟ କରେଇଦବା କାହା ଭିତରକୁ into the plasmid। the gene ଟା କଣ ହେଲା ଏଇ ଯୋଉ ଆମର desired gene ଅଛି the human gene is inserted in the plasmid and now the plasmid with gene is inserted in the bacterium once again। so basically we are having a bacteria ଆମେ ସେ bacteria ରୁ ତା plasmid କୁ ବାହାର କଲେ then ଏପଟେ ଏ ଯୋଉ human cell ଅଛି ତା ଭିତରେ chromosome ରୁ ସେ DNA ରୁ ସେ gene ଟାକୁ ବାହାର କଲେ then ଦିଟାକୁ ନେଇକି proper ratio ରେ cut କଲେ using same restriction enzyme and then ତାକୁ ଆମେ further ligate କରିକି insert କରେଇଦେଲେ ସେ plasmid ଭିତରକୁ and now the suitable bacteria ଭିତରେ ଆମେ insert କରିସାରିଲା ପରେ ସିଏ ଅବିକା grow କରିବା start କରିବ। ତ ଯେତେ ଥର ସେ bacteria ଟା reproduce କରିବ ସେ bacteria ଆଗକୁ ଯାଇକି ତାର cell duplication କରିବ replication process କରିବ ସେତେ time insulin ଟା ବି grow କରିପାରିବ।
एक रू 9 मार दे पासवर्ड पळा आके के जानछि
Done. Now focus। ତ ସିଏ ଏବେ ସେଥିରେ ଇନସୁଲିନ ବନେଇ ଯିବ ତ ବ୍ୟାକ୍ଟେରିଆ ଯେତେ ଗ୍ରୋ କରି ଯିବ ଆମର ସେ ଡିଜାର୍ଡ ପ୍ରୋଟିନ ସେତେ ଭଲସେ ଗ୍ରୋ କରିଯିବ। I have already talked about the three critical research area of biotechnology in the previous class in the morning class। The concept in the last one was like downstream processing। In the downstream processing, we are basically isolating the enzyme or isolating the protein and we are purifying the protein for the further use। ठीक से जिनस once a bacteria grow करिगला and से insulin production करिगा start करिगला then हमको से bacteria रो से insulin को काट दबार अछि ताको purify करिवार अछि and ताको यूज करिवार अछि। then but जेते इजीली मु तुमको कहि दिली कि एठु काढी दबा सेठु काढी दबा आगो काटी दबा जोड़ी दबा it is not easy at all। ए जिंस करिया पई बहुत टाइम टेकिंग हेतला and first in 1983 an American company Eli Lilly an American company Eli Lilly has first time ପାଇଁ achieve କରିଥିଲେ ଏ ଜିନିଷ to produce human insulin which is called as human। so in the next slide we are going to study about them in details। so if you have any doubt in this slide then you can ask me in the chat box। Dear participants, do you have any doubt till now? If you have any doubt, you can always ask me in the chat box। Okay, now let's move to the next segment। okay, so insulin consists of two short polypeptide chains, that is chain A and chain B, that are linked by disulfide bridges। remember ये first slide में क्या होता है discussion करते हैं, we are basically talking about the insulin and this insulin consists of two short polypeptide chains। what is polypeptide? Polypeptide means proteins, poly means many, and peptide bond which is present in between amino acids। ठीक है, we are having amino acid 1, amino acid 2, amino acid 3, amino acid 4 and all those amino acids are joined together by the help of peptide bond। now this is one peptide, monopeptide, dipeptide, tripeptide and the more peptides are called as polypeptide chain and this polypeptide is basically called as a protein। now what happened, this polypeptide chain has two chains, that is chain A and chain B, that are linked by disulfide bridges। both are linked by disulfide bridges। remember ये question भी आसीबो insulin जो अछि human insulin जो दीटा chain अछि A chain B chain, how they are connected? is it through hydrogen bond, is it through peptide bond, is it through nitrogen bond, is it through covalent bond, is it through peptide bond, disulfide bond? your answer is going to be disulfide bridges। ठीक अछि, जेम तमे पढिथिबो in zoology in the antibody structure ये जो Y shape रो antibody थिला the light chain और heavy chain थिले they are bound through the disulfide bridges। it is a disulfide bridges आसिला which is present here in the human insulin. Now, in mammals, the insulin is synthesized as a prohormone. Remember this. Then the beta toxin ne padhitile ki beta toxin the formation heichi. Initially it was produced as a protoxin, and then se protoxin ago jainki activate heitila in the mid gut epithelial cells of the insect. Yes or no? The protoxin e formation heitila ago jainki modify heichi, activate heichi. Exactly in human body, the insulin is produced as a prohormone. Prohormone means it is inactivated, it's not activated. It has all the three peptide chains, A peptide, B peptide, and C peptide. Then, so ये है ना prohormone. The pro जब mature हब, the prohormone when it gets mature hormone convert होती, then we need to remove the C peptide. The C peptide को remove कर देले, only A chain and B chain missing रहेगा. That is called as mature insulin. You can see in this structure in this figure, this is called as proinsulin. This entire segment is called as proinsulin. Yes or no? These are the disulfide bridges, and this is chain A, this is chain B, and here it is added with a C peptide add ହେଇକି ଅଛି. जब से C peptide को हम हटेई देले, ताको remove कर देले, free कर देले. Now this chain A and this chain B are linked through disulfide bridges. It is called as mature insulin. आकु हमें mature insulin बोली कहले. It needs processing to become a mature and functional hormone. And the prohormone contains an extra stretch called as the C peptide, and this is removed during the maturation into insulin. Manachi, in eukaryotes, when you are doing transcription in eukaryotes, then direct mRNA is not produced. If you remember, the students, I have already taught you in the transcription class that during our transcription in the eukaryotes, the DNA is not directly transcribed into mRNA. Rather, it is transcribed into a chain RNA, that is heterogeneous nuclear RNA. And inside that chain RNA, there are introns overlapping each other. That intron with the exon with the introns are basically the intervening compounds which has no role in coding, which do not code for anything. But the exons are the coding parts, coding sequences, yes or no? So ये जो exons मैंने वो किए cover कर रखा था, intron मैंने cover कर रखा. ଯେତେବେଳେ ଆମର ଟ୍ରାନ୍ସକ୍ରିପ୍ସନ ସରିଯାଇଥିଲା ଦେନ୍ ଆମେ କଣ କରିଥିଲେ ପୋଷ୍ଟ ଟ୍ରାନ୍ସକ୍ରିପ୍ସନାଲ ମଡିଫିକେସନ କରିଥିଲେ ଯାହାକୁ ଆମେ ଆରଏନଏ ପ୍ରୋସେସିଂ ବୋଲି କହୁଥିଲେ ହିୟର वी नीड टू क्लीव सिलेक्टिवली क्लीव କରିବାକୁ ପଡୁଥିଲା କାହାକୁ ନା ଯୋଉ ଇଣ୍ଟ୍ରନ୍ସ ମାନଙ୍କୁ ଦ ଇଣ୍ଟରଭେନିଂ କମ୍ପାଉଣ୍ଡସ୍ ଦ ନନ୍ କୋଡିଂ ସେଗମେଣ୍ଟସ୍ हम क्लिक କରିଥିଲେ and क्लिक କରିସାରିଲା ପରେ then exons are ligated ତ ଜଏନ୍ କରିଦେଲେ and here we get our desired mRNA structure. Like this is the protoxin. This is the proinsulin, which is containing three peptide chain. See, insulin is a hormone. Insulin is a protein. और जो protein है, protein is a polypeptide chain. Polypeptide means it is containing peptide bonds. Number of peptide bonds. It has three polypeptide chain: chain A, chain B, and chain C. So in the pro, there are three. 1, 2, 3, A, B, C. But in the mature insulin, you have only two. That is one small a and a large B. A small a and large B, and they are joined together through the help of this disulfide bridges. Altogether, it is having 51 number of amino acids. 51 amino acids are there, and among the 51 amino acids, 21 is present in chain A and 30 is present in chain B. So till now, if you have any doubt, you can ask me in the chat box. ଆଉ ଯଦି ବୁଝି ଯାଇଛ then we'll go forward. The participants, is there any doubt till now? Do you have anything to ask, or shall we move forward? Okay, so if you don't have any kind of doubt, then we can go forward. Now, see, this genetically engineered insulin in 1983, an American company, Eli Lilly, prepared the two DNA sequences corresponding to the A and B chains of human insulin. Okay, let me tell you, you need to remember this thing. The year is 1983. The name of the company is Eli Lilly, and it is basically an American company, and who have prepared the DNA sequences, two DNA sequences corresponding to chain A and chain B of human insulin. That is the game changer. This is the game changer. ସେମାନେ କଣ କଲେ କି ଆମେ ଜାଣିଲେ କି ହ୍ୟୁମାନ ବଡିରେ ଇନ୍ସୁଲିନ କେମିତି ବନୁଛି ଆଉ ଯୋଉ ପ୍ରୋ ହର୍ମୋନ ହିସାବରେ ବନୁଛି what is the pro hormone? Pro hormone basically कौन ही करे ना तमर सेठी A B C peptide ଅଛି and A B C peptide ଭିତରୁ ସେ କାହା ଉପରେ ଡିପେଣ୍ଡ କରୁଥିଲା ନା A ଆଉ B କୁ ରଖୁଥିଲା C ଟାକୁ ହଟେଇ ଦଉଥିଲା ବଟ୍ ଏଠି ସେମାନେ କଣ କଲେ ନା C କୁ ଯଦି ହଟେଇବାର ହିଁ ଅଛି ଫାଇନାଲି it is we are going to remove the C peptide so why should you make the C peptide ଯଦି ଆବଶ୍ୟକ ନାହିଁ କାହିଁ ବନେଇବା ତ ଆମେ A chain ପାଇଁ କି ଯୋଉ ଜିନ୍ ରେସ୍ପନ୍ସିବଲ ତାକୁ ନେଇକି ଇନସର୍ଟ କରିଦେଲେ ଗୋଟେ ଇକ୍ୱାଲାଇ ଭିତରେ B chain ପାଇଁ କି ଯୋଉ gene responsible ତାକୁ ନେଇକି insert କରିଦେଲେ another equilibrium ଭିତରେ and we have separately produced A chain and B chain, A peptide, C peptide, B peptide, and after formation of A and B ତାକୁ ଆମେ merge କରିଦେଲେ by the help of disulfide bridges. So this is going to happen in the part. So disulfide bridges ରେ ଆମେ ତାକୁ କଣ କଲେ ଯୋଡ଼ିଦେଲେ and they were introduced in plasmids of E. coli. So E. coli the plasmid हम तो E कर to produce insulin chain. Chain A and chain B are combined by creating disulfide bond to form human insulin. तो जो human insulin formation करिया पाई कि हमें कौन कहले chain A, chain B जीटा को disulfide बीजे से तब हम जोड़ी देले और ये जो bond बनी गला से bond को नहीं कि हमें finally जोड़ी चला पड़े human insulin production हैला और ये जो human insulin production है ची ताकि हमें कहले कि कौन humulin Humulin, it is called as humulin. What is humulin? Humulin is basically genetically engineered human insulin. Okay, so genetically engineered human insulin is called as humulin. And ये जो humulin बनिची, ये humulin टा केमिति बनिची? Humulin टी बनिची by the help of the E. coli. हम हेड chain A बनैले गोटे पटे रे, polypeptide chain B बनैले गोटे पटे, and D टाक हमें merge कर दे कि ताको हमें disulfide bond रे merge कर दे कि हम बनै देले human insulin humulin. This kind of thing was first developed in the American company Eli Lilly in the year 1980. Now, our next segment is gene therapy. So here we are going to discuss about the topic gene therapy. So basically, read in the screen what do you mean by gene therapy. First, learn the definition. So it is the method to correct a gene defect in the child or an embryo. So our defective gene को हमें कौन करते हैं change करते हैं. Change माने कौन है ताको हमें correct करते हैं. Yes or no? So there is a gene which is the defective gene. जो gene भी आगू जाएंगी किसी गोटे भूल प्रोटीन फॉर्मेशन करथिला रॉन्ग प्रोटीन फॉर्मेशन करथिला जो रॉन्ग प्रोटीन टी आगू जाएंगी गोटे बॉडी रे डिजास्टर करथिला सो इट इज द मेथड ऑफ करेक्शन of a defective gene in a child or an embryo. Here, the genes are basically inserted into a person's cells insert wale and the tissues to treat a hereditary disease and it compensates for one non-functional gene. So disease जो अच्छी पिला मैंने हम जो disease है what is disease? The word disease is basically called means dis-ease. Dis means discomfort, ease means comfort. So dis-ease, that is called disease. और जो डिजीज अच्छी डिजीज कैन बी congenital माने एटा जन्म inborn disease भी हमको which we have already inherited from our parents. So there are some mutation in the body, some gene mutations are there, some chromosomal aberrations are there. जोटा कि आगो जाएंगे हम बॉडी की डिफेक्टिव सो करते हैं जीन डिफेक्ट करेंगे. So by using this therapy, by using this gene therapy, we are able to correct the defective gene and we can replace the faulty gene by a normal or healthy and a functional gene. तो এই जो करेक्शन कथा कहला करेक्शन कौन कराई ना गोटे डिफेक्टिव जीन जो थिला डिफेक्टिव भी कहि परिबो और फॉल्टी भी कहि परिबो is being replaced by a normal healthy and functional gene. ଗୋଟେ ଜିନ୍ ଟା ଯୋଉଟା କି ଡିଫେକ୍ଟିଭ ବା ଫଲ୍ଟି ଥିଲା ତାକୁ ଆମେ କଣ କଲେ ରିପ୍ଲେସ କଲେ ଗୋଟେ ନର୍ମାଲ ହେଲ୍ଦି ଆଉ ଫଙ୍କସନାଲ ଜିନ୍ ରେ ରିପ୍ଲେସ କରିଦେଲେ दैट इज कॉल्ड द जीन थेरेपी. क्लियर? ओके, व्हाई शुड यू डू दिस काइंड ऑफ थेरेपी? कौन ପାଇଁ ଆମେ କରିବୁ? व्हाट इज द नीड ऑफ दिस थेरेपी एंड हाउ डू यू नो दैट द जीन इज डिफेक्टिव? और ଗୋଟେ एम्ब्रियो सेल केମିତି ଜାଣିବା? तो तमे जूलॉजी रो फर्स्ट चैप्टर सइला पहिले सेकंड चैप्टर रिप्रोडक्टिव हेल्थ अछि. In that reproductive health, you have heard about a term that is called as amniocentesis. And in that amniocentesis, you can withdraw the amniotic fluid from the child's body, from the mother's womb. You can derive the amniotic fluid, and by doing the karyotyping, you can see what are the chromosomal abnormalities or what are the chromosomal arrangement in that embryo. If you found any kind of chromosomal non-disjunction, that means it can lead to Down syndrome, it can lead to Klinefelter syndrome, it can lead to Turner syndrome. You can see whether there is monozomy, nolizomy, trisomy, whether there is abnormal chromosome. The chromosome number can raise to 47. The chromosomal number can raise to 40, can be reduced to 45. So what is basically happening there? So you can detect it by seeing doing the karyotyping of the amniocentesis. And after the amniocentesis, we got a detailed report, and we have also know by the Human Genome Project that which gene, which chromosome is responsible for which categories. We know all the 46 number of chromosomes, and जो 46 टा chromosome अच्छी 23 pair का function हमको जाना है କୋଉ ଜିନ୍ କାମ କଣ ସେଇଟା ବି ଆମେ ଜାଣିଛେ ଆଉ ସେଇ ଜିନ୍ ସିକ୍ୱେନ୍ସ ରେ କିଛି ଫଲ୍ଟ ଆମେ ପାଉଛେ. If you find any kind of fault, any kind of disorder in the gene arrangement, then we can correct that faulty gene or the defected gene by help of the gene therapy and or a normal functional healthy gene that we replace करें. This is the main motto of this part. If part to main motto होची. I hope you have understood till now. If you have any doubt, you can always ask me in the chat box. Now, the first gene therapy, if you see, the first gene therapy is done in 1990 and was given to a 4-year-old girl with adenosine deaminase deficiency. Remember, she is the girl, her name is Asanti De Silva. He is the first clinical gene therapy patient. जब से 4-year-old थिला ता बॉडी रे ए जीन थेरेपी प्रोसेस टाको क्यारी करा जायथिला. An American girl known as Santa De Silva, she was suffering from ADA deficiency and the name of the disease is SCID. She was suffering from SCID. And what is SCID? SCID is severe combined immuno deficiency. Severe combined immunodeficiency, it is a disease or it is a disorder which is due to absence of what? Adenosine deaminase deficiency. Then ADA is an enzyme. ADA enzyme absence re the lymphocytes basically harm has been. Our lymphocytes যদি harm হয়ে গেল তাহলে the immunity power পুরা কমে যাব. You know our body contains blood. এই যে blood আছে blood ভিতরে আছে তোমার plasma and your formed elements. Inside the formed elements you are having your RBC, WBC and platelets. RBC is a erythrocyte, WBC is thrombocyte and platelet. WBC is leukocyte and platelets are thrombocytes. ସେ ଯୋଉ ଲ୍ୟୁକୋସାଇଟ୍ ଅଛି WBC ଅଛି ତା ଭିତରେ ଗ୍ରାନୁଲୋସାଇଟ୍ ଦିଟା ପାର୍ଟରେ ଡିଭାଇଡ କରିଯାଏ। ଏବେ ଯୋଉ ଆଗ୍ରାନୁଲୋସାଇଟ୍ ଅଛି ସେଠି କି ଅଛି ଲିମ୍ଫୋସାଇଟ୍ ମୋନୋସାଇଟ୍ ସାଥେ ଆଉ ସେ ଲିମ୍ଫୋସାଇଟ୍ ଭିତରେ ଗୋଟେ ଟି ଲିମ୍ଫୋସାଇଟ୍ ଅଛି ଯେ କି ବେସିକାଲି ଆମକୁ କଣ କରୁଛନ୍ତି ନା ଡିଜିଜ କୁ ଡିଟେକ୍ଟ କରିବାରେ ପାଥୋଜେନ କୁ ଚିହ୍ନଟ କରିବାରେ ହେଲ୍ପ କରୁଛି। तो ये जो एडीए एडेनोसिन डीएमिनेज डेफिशिएंसी ହେଇଛି तो बेसिकली ये टी लिम्फोसाइट फॉर्मेशन को अटकेगा एंड द बॉडी टी लिम्फोसाइट डेफिशिएंसी होगा स्टार्ट होगा then naturally तर इम्यूनिटी वीकेन ହେଇଯିବ और जब हमर इम्यूनिटी वीकेन हो जाइए then हम लोग various kind of pathogens can easily target us can easily attack us so that we can suffer from severe combined immunodeficiency type of disease and a disease basically death ହେଇଯାଏ ସମ୍ଭାବନା ବହୁତ ଅଧିକ ଥାଏ। So by using this gene replacement therapy, by using this gene therapy, ये जो सेंट्रल डिसिल्वा नामक जेटी है जिसका बर्थडे है 1990 के फर्स्ट टाइम पे कि ये जो फॉल्टी जीन से डिफेक्टिव जीन को पढ़ाएला तो वो रिप्लेस कराएला और त्या जगह रे कहां को रखाएला गोटे नॉर्मल हेल्दी एंड फंक्शनल जीन को रखा so that ये जो एडेनोसिन डीएमिनेज फॉर्मेशन पुणी हे परिबो और जब ये फोन फॉर्मेशन हैला आ डिफिशिएंसी बंद हे गला ये बस सफिशिएंट फॉर्मेशन हैला देन तमे कोन जानि बेगी तो बॉडी रे एबे फोर्थ इम्यूनिटी पार्ट स्ट्रांग हे गला and she has no more to survive from this immunodeficiency syndrome. So now you can question one thing there students, you can question, sir, if the cause of the disease is ADA deficiency, so we can give ADA to the girl. What is ADA? Adenosine deaminase. Aminase means what? This is an enzyme. Then the lipase, ligase, then salivary amylase, then the helicase. These are enzymes. So the after enzyme deficiency, so we can provide an enzyme because enzymes are proteins. तो हमें enzyme protein बाहर बने बता बॉडी सर्वाइव दे देगा but कैसे नहीं दे पाएगा? Here is the question: for how long you have to provide that enzyme? The enzyme तुम्हें दो treatment by bone marrow transplantation or enzyme replacement therapy, injection of ADA is not completely curative. इन्हें completely cure कर पाएगा ना? ठीक है जी तब जो एंजाइम को रिप्लेस कर देबा एंजाइम रिपेयर जे हम एंजाइम दे दा बट किडनी एंजाइम जे करिवो गोटे नेचुरल गोटे नॉर्मल ह्यूमन बीइंग ये किडनी रे तार फिल्ट्रेशन होउची बॉडी रे जिंस को सीए जेमति सर्वाइव कर परिबो एंड द पेशेंट व्हिच डिपेंडेंट ऑन हेमोडायलिसिस द सी को सेवा सर्वाइव कर परिबो कि गोटे नेचुरल हार्ट जेमति फंक्शन करिबो गोटे आर्टिफिशियल पेस मेवर लागि हार्ट से फंक्शन कर परिबो कि मेजर डिफरेंस तो हमारे एडीए ର रिक्वायरमेंट अछि तमे एडीए प्रोवाइड कर देले बट ये क्यूरेटिव नुहे कंप्लीटली क्यूरेटिव नुहे अब बोन में ट्रांसप्लांट करा अगेन से पार्टी फोन आउट हो जाएगा आगो से असुविधा देखे ये रखितो ମାନେ बेसिकली बुझे को ट्राई कर पिला माने कि गोटा शॉर्टकट आउट लॉन्ग टर्म में फायदा पाइबा जेमते कि कोणिकिया घुसीबा आउ टेबल जो हमर कैलकुलेशन सिखिबा मल्टीप्लिकेशन सिखिबा बोथ आर डिफरेंट थिंग जदि गोटे पिला को क्वेश्चन पड़ला व्हाट इज 7 and 9 या 17 नुआ केते और आंसर से घुसी जे पणिकिया जो घुसी कहि परिबो सो 17 1 जेति 17 2 या 17 3 1 4 ना कहि 17 9 देगी केवल से बट से प्लेट मैक्सिमम 25 टेबल नहीं घुसी देबो जो हम स्कूल में देखे आछे 20 को 25 को कन्वर्जन लोग मान घुसता है बेसिकली बट इफ द पर्सन नो हाउ टू मल्टीप्लाई तब मल्टीप्लिकेशन जाना है then he or she no need to remember no need to memorize all those tables से जो थे भी जेतेबेले भी जेमेते क्वेश्चन पत्र के आंसर दे पारे बिकॉज़ ये जानि छी कि हाउ टू मल्टीप्लाई तो गोटे घोषा पाठटी बहुत कम कम जिन से अभी सर्वे कर परिबो ଯଦି ପାଠଟି ବୁଝି ପାରିଛି ସେ ଜିନିଷଟି ମଲ୍ଟିପ୍ଲାଏ ଶିଖିଛି ସବୁ ମୁଭମେଣ୍ଟ ସେଠି ଜିତି ପାରିବ। ଠିକ ସେମିତି ଯଦି ତମେ ଏଡିଏ ଡେଫିସିଏନ୍ସି ଅଛି ତମେ ଏଡିଏ ପ୍ରୋଭାଇଡ କରିଦେଲ ମାନେ ତା ରିକ୍ୱାରମେଣ୍ଟ ଥିଲା তুমি ଏଡିଏ ପ୍ରୋଭାଇଡ କରିଦେଲ ତ ସିଏ ଏବେ ଲଙ୍ଗ ଟର୍ମ ଯାଇକି ହେଲ୍ପ କରିପାରିବ ନାହିଁ। ये शॉर्ट टर्म में सीधा हेल्प कर परियो रिलैक्स दे परियो लॉन्ग टर्म जाके हेल्प कर परियो नहीं और ओनली गोटे बाटा थिला कोन that is the replacement therapy gene replacement therapy or gene therapy is the only part by which we can replace the faulty gene through a healthy normal gene. Clear? Any doubt till now? Dear students, do you have any doubt till now? See, now we are going to see a look in detail about the gene therapy and how this process is exactly happening. Now see, the gene therapy for AD deficiency collect lymphocytes from the patient's blood and grow in the culture. ये जो gene therapy जो काम करा होची, जो हमें AD deficiency पे देखिबा, they collect the lymphocyte. कहाँ collect करा जाए? Lymphocyte को from the patient's blood and grow in a culture. Next, we introduce a functional ADS cDNA. What is cDNA? Now, remember, cDNA stands for complementary DNA. Complementary DNA. ତା ବଡିରେ ଗୋଟେ functional ADA cDNA into the lymphocytes using a retroviral vector. Remember, in case of plant case रे vector किए थिला? In case of plant, the vector is agrobacterium tumefaciens. Agrobacterium tumefaciens is a vector in case of plant. But in case of animal, in case of human, the vector is a retrovirus. Or you can say adenovirus. Basically, I'm going to viral vector को use करके further transportation. Then they using a retroviral vector I'm use करेचे and they are returned to the patient and this should be periodically repeated by lymphocytes are not immortal because the lymphocyte immortal नुहंती हम एट कौन करते हैं तक हमें बारंबार दोचे. This is the patient. Sorry, spelling mistake is there. It is a patient PATIENT. ये patient रा हमार T lymphocyte को हमें बाहर करता बॉडी patient रा बॉडी रू patient रे शो करची patient is basically बहुत sad अछि दुखी अछि and you can see here से patient केते खुशी हो जाइछि तार बे disease भल गेला परे now he or she is happy. तो पेशेंट रो टी सेल को हमें रिमूव करले देन पेशेंट को रिमूव कर साला परे ताके हमें कोन करले ग्रोइंग द टी सेल इन द कल्चर मीडिया हमें लैबोरेटरी कल्चर तको रिमूव करले जो सेल ता बॉडी भीतर ग्रो करथिला तको हमें कल्चर प्लेट रानी ग्रो करेले देन हम डिटेक्ट करले से जो एडीए जीन ता थिला जो डिफेक्टिव एडीए जीन जोडा अबिका बंद हेला तको हम डिफेक्ट कर पाइले देन से जिन छोटा को हम इंजेक्शन करले इन्फेक्शन कहा करले हबे इंजेक्शन करले ह्यूमन एडीएस सीरेना तो ताजे पे इंसर्ट कर देले insert he sala pore now this T cell is inserted carries the human DNA cDNA तो पठाइला and सी एज कौन करसन लैबोरेटरी केस को जाई कि एम्प्लीफिकेशन करची एम्प्लीफिकेशन माने मल्टीप्लाई करिबा एम्प्लीफाई करची माने से आगो जाई कि मल्टीप्लाई करची और जब से मल्टीप्लाई करची नंबर ऑफ सेल इंक्रीज हेची and again we need to infuse back to the patient जो पेशेंट रो नेइथिले से पेशेंट रो कलेक्शन कला पर पठे देबे इट्स लाइक मो बॉडी रो गोटे पार्ट को कढा हैला देन कढाला पर डिफेक्ट जीन ताकू खोजा हैला ताकू रिप्लेस करा हैला गोटे हेल्दी जीन द्वारा हेल्दी फंक्शनल जीन द्वारा रिप्लेस करा हैला देन तापर फोन ताकू नै रहला इट्स लाइक टायर जो पंक्चर मरा होची गोटे लीक हो जाइए आम टायर को रिमूव कले देन से पंक्चर वाला पार्ट को डिटेक्ट कले एंड देन तक हम रिप्लेस कर देले गोटे नॉर्मल टायर सहित एंड अगेन हम तुम्हें इंसर्ट कर देले से पार्ट रे ठीक से बड़ा जिंस रे तो ये जो सैड थिला नाउ ही और सी इज बिकम हैप्पी ये लाइक अच्छा if ADS gene from marrow cell, bone marrow cell is introduced into the cells at early embryonic stages, then it could be a permanent cure. Why so? From an entrance examination, assertion written type of question is there. And in that assertion written type question, you will be given two questions, two statements are there. Statement one is called assertion, statement two is called as written. Now you need to see if both are correct or one is correct or one is incorrect. So you have to look for that. And again, if both are correct, then you have to also check whether the region is the correct explanation for the assertion or not. If the region is not correct for the explanation, then you have to choose another option. And if the region is correct, then you have to choose another option and you have to do this kind of things. From it is a technical just knowing the theory is not enough. You need to know the detailed analysis of that part. Yes or no? Just remember to the ki ei part ta kauna, sei part ta kauna, it's not going to help you. Because in MCQ question, data is sufficient. But in case of assertion reasoning wala question, the statement wala question, their analysis is important. So we should know the ADA gene तक हमें दौड़ से कहीं के deficiency कहीं होगी, periodically कहीं repeat करियो पड़ेगा. Why so? Because the thing is lymphocytes are not immortal. ये अमर नुहंती lymphocyte जो तुम कलेक्शन कर देला सेम lymphocyte considerably guarantee नहीं तेबने केबे से मरले जेम तुम जानिଛ RBC red blood corpuscles ताको लाइफ स्पैन इज ओनली 120 days तो जितने healthy RBC हेतले भी after 120 days it need to go to the spleen. Yes or no? Or the spleen is the graveyard of RBC. The spleen है कौन पे मसानी कहाँ पे मसानी हिची ना से जो RBC हो जाते after 120 days it need to retire हो से RBC जीवो and that RBC is being replaced by new RBC formation clear तुम देखि पारछो there Sachin Tendulkar can play cricket right now also but he is not playing for team India से कौन से format रे खेल ना but he is able to play cricket. Mahendra Singh Dhoni is still able to play cricket. But से IPL को छाड़ दिले को format रे खेल रहे हैं reason कौन ना ସେମାନେ जो age ही ଆସିଲାଣି तो they are replaced by the younger ones. Then so there is retirement age ଯେମିତି army case ରେ ହୁଏ कि बहुत जल्दी early retirement ଆସିଥାଏ फिर से बड़ा जिनिस एटी कौन ना lymphocytes are not immortal they are immortal तो ସେଥିପାଇଁ कि आपको बारंबार चेंज କରିବାକୁ पड़ता है but हाँ if you are doing this case in the early embryonic stage जब बहुत जल्दी कामटी कर देছো एम्ब्रियोनिक स्टेज में चेंजेस कर देছো ओके इट कुड बी परमानेंट क्योर इट कुड बी ये ठीक अछि इट मस्ट बी ये तो ही परिबो इट कुड बी अ परमानेंट क्योर जो अर्ली डायग्नोसिस तमे कर देছো स्टेज 1 रो तमे कब डायग्नोसिस करि पाइছো चिन्हट करिছো एंड तमे हेटी कि छोटे पदक्षेप निजे चेंज କରିବା ପାଇଁ ट्राई କରିছো देन इट इज गोइंग टू बी अ परमानेंट ସେଥିପାଇଁ कि हम छोट बड़े ना हैंडराइटिंग बहुत प्रैक्टिस करतो सेट ସେତେବେଳେ ଆମେ କଞ୍ଚା ମାଟି ଥିଲୁ ଆମେ ଯେମିତି ଯେତେବେଳେ ଗ୍ରୋ କରୁଥିଲୁ ଆମକୁ ଯେମିତି ସେପ୍ ଦିଆଯାଇଥିବ ଆମେ ସେମିତି ସେପ୍ ହେଇଥାଏ ତ ଯୋଉ ପିଲାମାନଙ୍କର ହ୍ୟାଣ୍ଡରାଇଟିଂ ଛୋଟବେଳୁ ଭଲ ହେଇଆସିଛି ପ୍ରାକ୍ଟିସ କରୁଛନ୍ତି ହସ୍ପିଟାଲ ଲେଖିବା ତ ଲାଟର ଷ୍ଟେଜରେ ଠିକ ହେଇଯାଏ ତ ଛୋଟବେଳର ପ୍ରାକ୍ଟିସ ନାହିଁ ଆଉ ସେ ଯୋଉ ମାନେ ଆମର ହ୍ୟାଣ୍ଡରାଇଟିଂ ଟା ଖରାପ ହେଇଯାଇଛି ତ ପରେ ଯେତେ ଟ୍ରାଏ କଲେ ବି ହ୍ୟାଣ୍ଡରାଇଟିଂ ଠିକ ହେଇପାରେ ମାନେ ଯଦି ବି ଠିକ ହବ ବହୁତ କଷ୍ଟରେ ହବ ଆଣ୍ଡ ରେଗୁଲାର ବେସିସରେ କରିବାକୁ ପଡିବ ତ ସେଇଭଳିଆ ଜିନିଷ ଯଦି अर्ली ଏମାନେ ଷ୍ଟେଜ କିଛି ଗୋଟେ ଚେଞ୍ଜେସ୍ ତମ ବଡିରେ ଆସିଯାଇଛି you know what to do, how to do, when to do. Then in that case, you are going to get benefit from that. It will be a permanent cure. In that case only. Otherwise, it is not going to help you further. So I hope you all got these points. If you have any doubt, you can still ask me in the chat box. Now, if you look at the molecular diagnosis, this is a conventional method, that is serum and urine analysis are not suitable for early diagnosis of diseases. As I have told you earlier, either I'm a blood test kariba by urine test kariba, these are not suitable for early diagnosis. Yes, serum test हमें करूछे माने something has already been happened. Serum test ହେଇगला, serum माने ହେଇगला blood. Serum माने होतो blood test करिबा. The serum test ହେଇଗଲା, urine test ହେଇଗଲା, ये analysis are not suitable for early diagnosis. We can diagnose, but not these are early diagnosis. ଯଦି ତମ blood ରେ ଗୋଟେ infection ଟେ ମିଳିଛି, ମାନେ be sure ସେ infection ତମ body ରେ ହେଇ ସାରିଛି. We cannot change this scenario. Already it has been occurred in the body, and it is possible by techniques like recombinant DNA technology, PCR, and ELISA. So these are the technique by which you can only diagnose a pi. એટલે મને ବୁଝ ଭଲସେ କି something is going to come to your body. ଯେମିତି asteroid ହେଇଗଲା, ଗୋଟେ asteroid ଟେ ଖସୁଛି ଉପରୁ, ତମେ ତାକୁ ଦେଖିଲ ପଢୁଥିବାର ଦେଖିଲ, ତ ଅଲରେଡି ସେ ପଳେଇ ଆସିଲାଣି, ତମେ ଆଉ ଚେଞ୍ଜ କରିପାରିବନି। ବଟ୍ ଯଦି ଆସିଲା ପୂର୍ବରୁ ତମେ ପ୍ରେଡିକ୍ଟ କରିଦେଇଛ, then you can get prepared for this. ଯେମିତି ବର୍ଷା ହେଇଗଲା, ବର୍ଷା ହେଲା ପରେ ରେନକୋଟ ପିନ୍ଧିବା ଆଉ ବର୍ଷା ହବାକୁ ଅଛି ବୋଲି ପ୍ରେଡିକ୍ଟ କରିକି ରେନକୋଟ ପିନ୍ଧିବା, there is a difference between them. Yes or no? ବର୍ଷା ହେଲା, ସମସ୍ତେ ଦେଖୁଛ ବର୍ଷା ହଉଛି, then ତମେ ଗାଡି ଅଟକେଇଲ, ରେନକୋଟ କାଢିଲ, ରେନକୋଟ ପିନ୍ଧିଲ, it is a different scenario. But if you are sure enough कि जो अभी कंडीशन अच्छी आकाशर से कंडीशन देखी तमे जानी पारछ कि बादल माने एमती अछंती and agu it is going to be a big rainfall and you are prepared enough so that तुम रेनकोट पिंधि घरु बाहरिछ, that is called as prevention is better than cure. तो बाटरे ठिया कि तरतरी कि रास्ता मझिरे रेनकोट पिंधवा अपेक्षा घर जो तुम ऑलरेडी डायग्नोसिस कर देइछ, प्रेडिक्शन तुम ठीक अछि कि आगो वर्षा हवा को जाउछि एंड यू आर प्रिपेयर्ड फॉर द सेम, देन दैट इज अ डिफरेंट सिनेरियो. तो सही पार्ट दैट कहउछि पिला माने कि the conventional method, that is serum and urine analysis, are not suitable for early diagnosis of diseases. It is possible by techniques like recombinant DNA technology, that is PCR, polymerase chain reaction, or ELISA, that is enzyme linked immunosorbent assay. These are the techniques, these are the things which are going to be helpful. Clear? I hope तुमने बुझी पारछो. If you have doubt in this segment, you can ask me in the chat box. I am always there to answer to all your questions. Now, let's quickly see a very quick analysis about the PCR, which stands for polymerase chain reaction. The presence of a pathogen is normally suspected only based on symptoms, and by this time, the concentration of pathogen is already very high in the body. So you can look at this PPT, you can look at the slide. If you want, you can write it down in your note copy. And if you have doubt, then you can ask me. See, we're talking about okay. These two more slides. Then we are going for the question answer round. So just write it down in a copy, and then we will discuss. Today participant, have you written this thing? समझ लिखी दिलो पार्ट का. So we are basically talking about PCR. Now this PCR stands for polymerase chain reaction. And what is this polymerase chain reaction? It is basically an in vitro DNA replication mechanism. You know, DNA is being replicated inside the nucleus during the phase stage of cell cycle. Done. So basically, DNA replication is in vivo. Inside the cell, inside the nucleus. If we can replicate the DNA outside the body, that means in vitro DNA replication, it basically leads to in vitro DNA replication basically leads to what? Basically leads to PCR, that is polymerase chain reaction. You are going to amplify the copy of DNA. मैं DNA copies को amplify करते हैं. Now, the presence of a pathogen is normally suspected only based on symptoms. Usually I'm देखते कि हमें केवल डॉक्टर पाखु जाते हैं. मैंने when we go to a doctor to visit a doctor, बहुत rarely हम डॉक्टर पाखु जाओ देखने ପାଇଁ कि otherwise हम symptoms आसिला, we know these are the symptoms, we go to a chemist, we get some medicines and just eat it. କିନ୍ତୁ କଥା बहुत खराब हुए 3 दिन 4 दिन 5 दिन लगातार ज्वर है और लगातार सिम्पटम टे रहुची तुम क्योर होने देन यू गो टू अ डॉक्टर एंड देन डॉक्टर से टाइम में तुम डायग्नोसिस करबा पई से कहिबे कि यू गो एंड डू दिस टेस्ट. हम ज्वर है सपोज 3 दिन 4 दिन लगातार ज्वर है कि रहिला बट ज्वर भलो होनी हम मेडिसिन खाइले देन डॉक्टर पास गला बेलको डॉक्टर हमको एडवाइस करिवे यू गो एंड डू दिस काइंड ऑफ टेस्ट. तुम ए टेस्ट करो, तुम से टेस्ट करो, ब्लड टेस्ट करो, यूरिन टेस्ट करो, वायरल फीवर हिच ना हेनी, मलेरिया टेस्ट करो, टाइफाइड देखो. This is the but remember, by this time already the concentration of the pathogen is very high in the body. That means already by this time the concentration of pathogen has been increased in the body. Body bhitire the pathogen enhancement increase kar. However, very low concentration of a bacteria or virus can be detected by amplification of their nucleic acid by PCR. That is the game changer here. That means what happens here कि जर हिला परे ताकू प्रेडिक्ट करबा तो बहुत इजी अछि. हम जानि पर डायग्नोसिस करके बट हवा पूर्व जब प्रेडिक्ट करि परछे then that is the game changer part. Then for example, this is my phone. I have the phone. Let's say in front of me there is a wall. In that wall something is written which I cannot see in my naked eye. But if I can see, I can read it. I can't read. मुझे पढ़ी पारुनी कौन लिखा है बोली. So मु सेठी जाएंगी पढ़ीबा अपेक्षा I have my phone and I can open my camera and zoom it. तो सेठी न जाएंगी दूररू I can predict it by using my phone. Yes or no? ठीक सेमती सेठी गले तो मु देखि परिबी पढ़ी परिबी सेठी जाइथिली पढ़िया पे लिखा है यूजलेस गोटे वर्ड लिखा है तार किछि मीनिंग नथिला. तो मु एते बार ट्रैवल करी गली पहुंचली पढ़िली रिटर्न करके आसिली which is no use. And if I have my phone I can stand here and I can zoom the photo and I can see whether there is something written or not. तो मु तो जानि परिबी से पार्ट को. So that is the basic difference. Okay. So if I have this PCR, then it is the game changer because here very small amount concentration of bacteria or virus can be detected by amplification. I can basically amplify. मतलब amplify कर देबी ताको magnify कर देबी देखिए भाई कौन insert है. That is the game changer here. Remember, the DNA is being replicated inside the nucleus basically, but during the stage of cell cycle, which is in vivo process, but in vitro DNA replication is called as PCR, polymerase chain reaction. ଆଉ ଏ PCR ଦ୍ୱାରା ମୁଁ ଗୋଟେ ଭାଇରସ ହଉ ବା ଗୋଟେ ବ୍ୟାକ୍ଟେରିଆ ହଉ ଆମ କନସେନ୍ଟ୍ରେସନ କୁ ବହୁତ କମ ଆମାଉଣ୍ଟ ଥିଲେ ବି ତାକୁ ଧରି ପାରିବି, ଟ୍ରେସ୍ ଆମାଉଣ୍ଟ କୁ ଧରି ପାରିବି, ବିକଜ ସେ ଆମ୍ପ୍ଲିଫିକେସନ କରିଦଉଛି। The name of this machine is called as thermal cycler. ठीक है, PCR machine thermal cycler, and these are microcentrifuge tube. ताको हमें MCT tube बोली कहता हूँ। এই MCT tube रे हम sample को हमें load करके हमें PCR machine ने, this is a PCR thermal cycler plate method, आज हम रखेंगे temperature हमें we can say the temperature from here और तीनटा step अच्छी स्टेप ऑलरेडी बायोटेक पार्ट रे पढ़ी जाइथिबो। जो तीनटा स्टेप रे हमें कहा विषय कथा होचे, लाइक फर्स्ट एला ताको हमें भांगुचे, जो डीएनए डबल हेलिक्स स्ट्रक्चर थिला ए डबल हेलिक्स स्ट्रक्चर को हमें ताको सिंगल स्ट्रैंड करबा, सो दैट से सिंगल स्ट्रैंड हिला परे ताको हमें कहा बिना डीनेचुरेशन ताको भांगिले, देन ताको हम लिया प्राइमर ताको ऐड करबा, दैट इज कॉल्ड एज एनिलिंग, प्राइमर ऐड हबो एंड देन तापर से कोन हबो मॉडिफाई हेके पूरा चेन एक्सटेंशन प्रोसेस चलबो। तो आगे हमें ताको डीनेचुरेशन करबा, देन हमें एनिलिंग करबा, देन हमें एक्सटेंशन करबा, माने डबल हेलिकल स्ट्रक्चर को हमें डिवाइड कर देले, सिंगल स्ट्रैंड कर देले, देन प्राइमर को ऐड कर देले एंड देन प्राइमर एक्सटेंड करिवो इन द डायरेक्शन ऑफ 5 प्राइम टू 3 प्राइम डायरेक्शन में एक्सटेंड करिवो, दैट इज द सिनेरियो। Clear? So this is all about little bit idea about PCR, which comes inside the applications of biotechnology in the molecular diagnosis segment. What are the uses of PCR? It is used to detect HIV. What is HIV? Human immunodeficiency virus in suspected AIDS patient. And what is AIDS? AIDS is a disease that is acquired immunodeficiency syndrome, which is basically carried out by the help of this virus, the retrovirus. HIV is very, very dangerous retrovirus. How it is category inside the STI, sexually transmitted infection or STD category. To detect the mutations in gene in cancer patients. You know what is cancer? Cancer what is her meaning? Cancer is nothing but uncontrolled cell growth. Uncontrolled cell growth or uncontrolled cell division. ठीक है जी, ये को जीन माने रेस्पोंसिबल ए जो ऑन्को जीन्स माने अच्छा कैंसर का जीन्स ऑन्को जीन्स जो एठी के जो म्यूटेशन हे जाई छी, व्हाट इज म्यूटेशन? एनी काइंड ऑफ सडन चेंज इन द जेनेटिक अपीयरेंस, whether due to addition or due to deletion or due to translocation or due to repetition or due to insertion, any kind of thing जरी किछि गोटे सडन चेंज हे छी, से चेंज ता आगो जाके म्यूटेशन ने कन्वर्ट हेतिबो, आ म्यूटेशन हवा द्वारा सेल द कंट्रोल ऊपर हरै बसिबो, सेल डिवीजन टे जोडा 24 आवर रे 1 सेल 2 सेल हेताता they quickly double hejibo. A single stranded DNA or RNA tagged with a radioactive molecule that is called as a probe is basically hybridized to its complementary DNA in a clone of cells, and it is followed by detection using autoradiography. Then autoradiography process we detect karipa and detect kariki hum seya kothe use kariba. The clone having mutated gene will not appear on photographic film because the probe will not have complementary with the mutated gene. तो से जो probe टी mutated gene से bind हे परिबो नै, ता पाइनो ता प्रकार किछि information नै, तो सी हमकु सेठी किछि शो करिबो नै, band शो करिबो नै, हम आकु देखि जानि परिबा। In a higher studies classes, if you go for BSc, MSc, PhD, then you will practically handle this kind of instrument and you will get to know a lot more about them. What is PCR? What is RT-PCR? How many types of markers are there? What is REPD? What is AFLP? Then what is RFLP? In different parts of PCR. Now, coming to the question and answer segment, let's quickly start. I'm waiting for your answers. So, dear participants, I'm requesting all of you to kindly unmute and answer to this question, or you can also use your comment box or chat box to reach me and send me the answers. To partner Porja, you can answer me in the chat box. Like Let me read the question for you. The first human hormone produced by recombinant DNA technology is called dash dash. It's a very easy question. Come on, answer it. The first human hormone produced by recombinant DNA technology. What is the name of that hormone? Who will tell me? Dear participants, please awaken. What is the name? Whether it is estrogen? No. Whether it is progesterone? No. Whether it is thyroxine? No. What is the right answer? The right answer is yes, Pragya. Correct answer. Purvi. Correct answer. Answer is insulin. Insulin is the correct answer. Congratulations. Now the second question. Pick out the incorrect statement. Here have four statements. You need to find out which one is incorrect. Like the insulin consists of two polypeptide chains that are linked by hydrogen bond. Hydrogen bond, no. These are picked by sulfide bonds. ଏବେ ହିଁ କହିଥିଲି insulin ଟା କୋଉଥିରେ ଅଛି disulfide bridge ରେ bind ହେଇକି ଅଛି. Disulfide bridge. Then disulfide bridges you have a binding ethila. Insulin used to manage adult onset diabetes. Yes, it is correct. It is possible to produce human insulin using bacteria. Yes, it is correct. That is called as Humulin. American company Eli Lilly has done this. And in mammals, the insulin is synthesized as a pro-hormone. That is pro-insulin. Yes, it is correct. The question पचारती which one is incorrect? The incorrect answer होची this one. Because a hydrogen bonded बोले लिखा है. Then the right answer is option A, and the answer is insulin consists of two polypeptide chains that are linked by disulfide bridges. Next, the pro-insulin contains an extra stretch called as gas. Whether it is A peptide, B peptide, or C peptide or D peptide. Come on, answer this is very simple. Yes, Pragyan, the answer is correct. The answer is C peptide. Yes, so the C peptide टा से प्रो हार्मोन ने थाए जोटा को हम आगु जाएंगे काट ठीक अछि जो हार्मोन हम इंसुलिन देखितले आ पाखरे ए चेन अछि आ पाखरे बी चेन अछि एंड आ पाखरे सी चेन अछि जे ए और बी को कनेक्ट करी रखता दिस पार्ट इज द सी चेन बेसिकली तो ये मैच्योर इंसुलिन जला हबो ए सी पार्ट टा को हम एटो कट कर देबा कट कर सला पर जो ए और बी रहइबो दीज आर बेसिकली बाइंड विथ द डाइसल्फाइड bridges. Process. Now the right answer is yes, the C peptide. Now question number 14. The first American company that prepared two DNA insulin and introduced them in a plasmid of E. coli to produce insulin chain was called. You just need to name the company, American company, whether it is Biomarine, Amgen, Eli Lilly, Illumina. What is your answer? Let's see. Yes, the answer is Eli Lilly, correct. Pragya and Purvi both have given the correct answer. Eli Lilly is the American company who was first time in the year 1983. First time they have uh, produced or the first insulin humulin in the company. Next, the first clinical gene therapy in 1990 was given to a 4-year-old girl, and the girl is suffering from which syndrome? Whether it is sickle cell anemia? No. Phenylketonuria? No. Myotonic dystrophy? Yes, again no. And the correct answer is adenosine deaminase deficiency, which is called as ADA. Yes. ADA deficiency is the correct answer. A is the correct answer. Pragya and Purvi who both have given the right answer. Now, the genetic defect, the adenosine deaminase ADA deficiency may be cured permanently by this. So, permanently can be cured by administering the adenosine deaminase through injection. Injection button permanently cure ହେଇପାରିବ ନା bone marrow transplantation permanently cure ହେଇପାରିବ ନା enzyme replacement therapy permanent cure ହେଇପାରିବ ନା introducing gene from marrow cell producing ADA into the cells at early embryonic stages. जो चारटा को पार्ट से क्योर तो ହେଇପାରିବ ହେଲେ क्वेश्चन पड़े permanently cure केमती हबो permanently cure ହେଇପାରିବ. Purvi saying answer B, but no Purvi. Sorry to say. Yes, Pragya. Pragya has given the correct answer, and the right answer is option D. Need to remember चारटा को पार्ट से क्योर तो ही परिबो हेले परफेक्टली क्योर परमानेंटली क्योर कोठी ही परिबो in the last segment, that is introducing gene from marrow cell producing ADA into the cells at early embryonic stage. So हमें early embryonic stage तक हमें क्योर करी परिबा, then it is going to make a change. Now the last question of today, that is PCR can be used for dash. PCR को हमें केमती यूज करी परिबा to detect HIV in suspected AIDS patient, to detect gene mutation in cancer patient, to identify many genetic disorder, or all of the above. And you know, all of the above is the only magnet which stretches all the participants at attention to this question. The correct answer is option D. Yes, Pragya has also given the correct answer. The right answer is option D. And PCR can be used for to detect HIV, suspect AIDS patient, to detect the gene mutation, or to identify many genetic disorders. The correct answer is option D. So thank you for your kind attention. Thanks for the and hereby we have come to end of this part. Yes. So it's for your kind attention, dear participants. You can log in to website by entering the QR code printed on your admit card. And your password is initially 123456, which you can change after first login. And you need to use this portal for notes, for PDF, to ask questions, post your answers in writing with audio, video. So on behalf of BHSC Odisha Online Academic Support Program, I thank you all of you for your kind attention, and we definitely meet again in the next